Good morning, students. Now coming to sexual reproduction in bacteria. Already you know there is no the true sexual reproduction in bacteria, but genetic material it is transferred from one bacterium to the next bacterium by some special methods. Now such methods are called recombinations. Already you know recombinations are three types. One is conjugation, second one transformation, third one transduction. Already explained the type method of conjugation in previous class was discovered by Gender E.L. Thornton and Lederberg in Escherichia coli. Now coming to the second method of reproduction, this is transformation. Okay, transformation. This is the second type of recombinations in bacterial cells. And next one, third one, this is transduction. Transduction. This is third type of recombination, means sexual reproduction in bacteria. Now coming to the second one, transformation. Students, this type of transformation was first discovered by scientist Frederick Griffith. Okay, Frederick Griffith, discovered by scientist Frederick Griffith. But he discovered this type of method, this sexual reproduction in bacteria in so he discovered this method of sexual reproduction especially in Streptococcus pneumoniae bacterium. Streptococcus pneumoniae. Okay, Streptococcus pneumoniae, very important. Streptococcus pneumoniae, this is one of the parasitic bacterium and it is the causal agent of bacterium for pneumonia, disease pneumonia. So this method of transformation was discovered by Frederick Griffith in Streptococcus pneumoniae. Students, this transformation it involves the uptake of naked segments of DNA present in atmosphere and incorporation of this segment of DNA into the recipient chromosome, that is in a heritable form. Okay, for example, this is bacteria, this is recipient bacteria. Okay, this is recipient bacteria. Now this is the genetic material present in the recipient bacteria, and these are the fragments of DNA present in bacterial surroundings, segments of DNA present in surrounding of this bacterial cell. So in this method, after the death of some bacteria, the genetic material present in this bacteria, that is DNA, it is broken into several segments. It is broken into several segments like this. Actually, this is the DNA present in DNA present in bacterial cells. But here, some bacterial cells are undergo death. After the death of some bacteria, the genetic material DNA present in this bacteria, it is broken into several small segments. Now these segments, they are present in the surroundings. At the same time, this is the recipient bacteria. Now it takes the segments of DNA from the surroundings. Okay, some bacterial cells, they take up the segments of DNA present in their surroundings and now these bacteria, it incorporates these segments into the recipient chromosome. Okay, these bacterial cells, they take up some fragments present in surroundings. Now these fragments, it is incorporated into the recipient chromosome like this. It is incorporated into the recipient chromosome. This is incorporated DNA segments. In this way, some bacterial cells, after the death of some bacterial cells, the DNA present in this bacteria, they are broken into several segments. Now they are present in the surroundings. Now some bacteria, they take up the segments of DNA from their surroundings and incorporates into the recipient chromosome. Now this DNA segment, it is mixed with the bacterial DNA. Now it becomes the inheritable in nature. Inherited, very, very important. Now this DNA fragment, it is incorporated into the recipient chromosome in heritable form. Now this DNA, it is also multiplied along with the bacterial DNA, and it is also continued from one generation to the next generation. In this way, due to the incorporation of some extra segments of DNA, means some extra genes into the recipient, these recipients, they also express some new characters which are expressed by the sequence of new DNA. In this way, after the death of some bacterial cells, their genetic material is broken into several segments and are present in surroundings. Now some bacteria, they take up these DNA fragments from their surroundings and it is incorporated into the recipient chromosome in heritable form. Now this bacterial DNA with a new sequence of DNA, it is also continued from one generation to the next generation by binary fission. Then is also express some new characters. This type of sexual reproduction present in bacteria, this is considered as transformation. Was discovered by Streptococcus, discovered by Frederick Griffith in Streptococcus pneumoniae. Okay. At the same time, now coming to the third type of sexual reproduction in bacteria, that is called transduction. Very, very important. Transduction. Students, you know, this transduction was discovered by scientist Gender and Lederberg. Gender and Lederberg, discovered by two scientists, Gender and Lederberg, in bacterium Salmonella typhimurium. Salmonella typhimurium. Okay, Salmonella typhimurium. This is one of the bacterial cell which is responsible for typhoid agents. Salmonella typhimurium. Students, you know, some of the viruses, they infect the bacterial cells. They are parasites on bacterial cells. The viruses which attack the bacterial cells and which infect the bacterial cells, such viruses are called bacteriophages. They are called bacteriophages. The viruses which attack bacteria, which infect the bacteria, such type of viruses are called bacteriophages. Okay, for example, this is one bacterium and this is bacteriophage. Now this is another bacterium and this is another bacteriophage. These type of bacteriophages, they infect the bacteria, attack the bacteria simultaneously. So while these bacteriophages infect the bacterial cells, the DNA present in bacteria that may enter into this virus. Now when it attack the another bacterial cell, the DNA which is received from this bacteria, it is transferred into the next bacteria. Okay, when this virus it attack one bacterial cell, the plasmid DNA is present in a bacterium may be entered into the virus. While it attack another bacterial cell, such plasmids of these bacteria may release into the next bacterium. So this is the transfer of genetic material DNA from one bacterium to the next bacterium through bacteriophages. That is called this is transduction. Very important. This is a phase mediated transfer of DNA between the bacterial cells. Okay, so the transfer of genetic material from one bacterium to the next bacterium through bacteriophage viruses, that is considered as transduction. It is called by Gender and Lederer, and in Salmonella hypomyelium. This is the transfer of genetic material from one bacterium to the next bacterium through viruses. What type of viruses? Bacteriophage viruses. What are bacteriophage viruses? The bacteria that the viruses that attack bacteria, infect bacteria, they are considered as bacteriophages. First described by Gender and Lederer in Salmonella hypomyelium. Students, now coming to a small topic. This is problems on bacterial cells. Problems and bacterial cells. Students, problems, there are some problems in conjugation of bacteria and binary fission of bacteria. First, coming to a symbolic example of a problem on conjugation, that is one bacterium, okay, in a medium, very important, in a medium, okay, in a medium, there are 500 F plus bacterial cells, okay, and 750 F minus bacterial cells, okay. There are 500 F plus cells, 750 F minus cells in a medium, okay. Here there are 500 plus cells and 750 minus cells, and these cells completed conjugation once. Okay, these cells completed conjugation once. Okay, now coming to the question: how many F plus cells and how many F minus cells are present in such a medium? How many F plus and F minus cells present in a medium? Okay, this is the question. Students, in a medium there are 500 F plus cells and 750 F minus cells, and all these plus and minus cells they completed conjugation in one time. And what is the total number of F plus cells and F minus cells present in such medium? Students, this is a small problem on the concept of bacterial conjugation. You know the concept of bacterial conjugation, plus and minus cells come close together. One plus cell, one minus cell come close together. They establish a conjugation tube. Now plasmid present in F plus cell duplicates into two copies. Now one copy is migrates to the minus cell. At the end of conjugation, minus cell that participated in conjugation becomes a plus cell. So here 500 plus cells, they are conjugated with 500 F minus cells. Okay, 500 cells plus cells conjugated with 500 minus cells. Now these 500 minus cells at the end of conjugation, they are converted into plus cells. So total plus cells, total F plus cells. Basically 500 F plus cells are present in medium. Now these 500 plus cells conjugated with 500 minus cells. Now these 500 minus cells are converted into plus cells. So 500 plus cells are also converted from minus cells. Total plus cells 500 plus 500. This is equal to 1000 F plus cells are present in medium. Okay. But what about total number of minus cells, F minus cells? Basically F minus cells are 750. Among these 750 minus cells, 500 cells are participated in conjugation and are converted into plus cells. Remaining cells are minus cells. So among these 750 minus cells, 500 plus 500 F minus cells are converted into plus by conjugation. Only 250 minus cells are there. Okay, 200 minus. Total minus cells, initially minus cells are 750, but 500 minus cells are converted into plus cells. Minus 500 is equal to 250 F minus cells. They are present in the medium. This is a small problem. Students, the main statement in this conjugation, that is, at the end of conjugation, minus cell it becomes a plus cell. So basically 500 plus cells are there. By conjugation, 500 minus cells are converted into plus cells. 500 plus 500, 1000 plus cells are there. And basically 750 minus cells are there. But 500 minus cells are converted into plus cells by participating conjugation. Remaining 250 minus cells are there. Totally 1000 plus cells and only 250 minus cells are present. Students, now coming to another problem. This is one bacterium is continuously participating binary fission in a medium. Okay? In a medium. One bacterium, okay, continued binary fission for 2 hours. Okay, 2 hours. And what is total number of bacteria found in medium? Okay, this is a question. Students, every bacterial cell it divides by binary fission for every 20 minutes once. That is called generation time. Okay, how much time is required to complete the process of binary fission in one bacterium? 20 minutes time is required to complete the process of binary fission. Means for every 20 minutes one bacterium is multiplied into 2 bacterial cells. Means in every binary fission the number of cells would be doubled. Okay, Takes 20 minutes for one binary fission. How many binary fissions are completed within 1 hour? 3 binary fissions are completed within 1 hour. Okay, and that is for 1 hour 2 power 3. 2 power 3. But binary fission it occurs at continued 2 hours. What is the total number of cells present in such a medium? Every bacterium takes 3 binary fissions for 1 hour. For 2 hours, bacterium it multiplied 6 times. That is 2 power 6. 2 power 6. Okay. This is 2 power 6. In one bacterium multiplied into 2 bacteria, 20 minutes. Now 2 is multiplied into 4 in 40 minutes. 4 is multiplied into 6, 8 in 1 hour. And 8 is 1 hour 20 minutes. 8 is divided into 16. 1 hour 40 minutes 32. 2 hours 64 cells are formed. Okay, 1, 2, 3 divisions for 1 hour. 1, 2, 3 divisions for the next hour. Total 2 power 6. That is equal to 64 cells are formed. 64 cells are formed in 2 hours. Okay, 64 cells are formed in 2 hours. That is 2 power 6. For example, one bacterium it is continued binary fission for 2 hours 20 minutes. How many cells are formed in this 2 hour 20 minutes? In 2 hours 2 power 6, 64. In 2 hours 24, they complete one more binary fission in this extra 20 minutes. That is 2 power 7. That is equal to 128. That is a simple problem on binary fission. So, this can be expressed as 2 power n. 2 power n. What is the meaning of n? N is equal to number of generations. Okay, this is a number of generations.